My name is Timothy James Waller, and I'm with Rutgers Cooperative Extension, based out of Cumberland County. My main duties are dealing with commercial nursery production. And given that I'm one of the newer members to Cooperative Extension, we are doing this Lunch and Learn series to give everyone an idea of who I am, both academically and personally, as well as some of my ideas moving forward. So with that, today's talk is going to go into my personal and academic background to give people a feeling of who I am as a person and the tools that I'm capable of utilizing. We're going to look at my previous research and go into my dissertation topic, as well as some of the other more notable research that I've conducted, as well as some of the other things I've picked up along the way in doing a lot of field trials. And then moving forward, I began with the department January 1st, 2020. And since then, I've been able to identify some needs, both research and educational, that will be really important in developing a viable commercial nursery production extension program. So this is a larger picture of myself, my characteristic yellow glasses. We're going to go into a little bit about my background. As you can see here, that blueberry and cranberry research and extension, I've spent the last 8 or 9 years, almost 10 years working in blueberry and cranberry. This is in Chatsworth, New Jersey, in the heart of the Pine Barrens. And while I was there, I worked on blueberry, cranberry, and grape. And I was both a lab and field technician at different times, but I was also in charge of supervising all the personnel, up to 10 people at any given day, and running 15 different jobs at the same time. I'm a licensed pesticide applicator. I have been for a number of years now, and I have pulled that trigger more times than I care to remember. In addition to these day-to-day activities, I was also a graduate student taking classes in New Brunswick and coming back down to the Pine Barrens to conduct my research. I also helped out with a lot of different projects and a lot of different groups. My main background is fungal phytopathology, so plant pathology, but I've worked with everything from breeding to entomology to looking at genetics, as well as field chemistries or helping out, harvesting, you name it, I've done it at that station. One of my projects, I got very into looking at molecular ID, ITS sequencing data of different pathogens so we could identify what they are. And through collecting field samples and grower samples, I've gotten pretty good at diagnostics, and I at least know which direction to head. So that's a skill set I've developed over the years. And I have to note, I was funded through a commodity group, the William S. Haynes Senior Endowed Cranberry Research Fund, put forward a lot of money to help me look at different fruit rotting diseases in cranberry, as well as allowing me to be boots on the ground at the station. In addition to my academic background when it comes to plants and pests and problems, 2 of my best friends are growers in the area. They do diversified farm stand produce, all kinds of mixed veggies, cut flowers, different herbs, growing up transplants on a small nursery scale to sell at the farm markets. And because of that, I've dealt with every type of problem. Just today, I had 2 different calls about hand sanitizing stations and what nutrient load one of my friends thinks his tomatoes should be getting at this time of year. In addition to working with my grower friends, I spent a number of years working for a tree service where I was the grounds foreman, climbed occasionally, and really ran day-to-day operations, driving the trucks, getting chippers out, you name it. So I can handle some pretty high-stress situations as well as personnel, and I can yell pretty loud when I need to be. But I've been working since I was 14 years old. We didn't get an allowance. It was, if you wanted money, you went for it. So I've also been a conveyor mechanic. I've done working on diesel engines. I learned how to weld hanging upside down in a mezzanine, different conveyor belts, all kinds of things that would go on in a distribution type warehouse. I've also done custodial, laid and waxed floors. I've been a carpentry assistant. I'm very keen on building things. And one of my first jobs growing up was fine dining. And although not academically related, it showed the importance of speaking to people with sincerity and being kind and getting the job done in a quick and timely fashion. One of my more interesting backgrounds is that I've worked with parrots for most of my life at this point. I've done parrot training and grooming, and believe me, when a beak of that size goes through your hands, it's something else. Some other notable hobbies, I'm a huge student of learning. I like to know how to do a lot of different things because, especially in the plant sciences and the extension world, you get hit with questions from every different angle. So the more you have to pull from, the better off you're going to be when it comes with dealing with real world problems. I'm a big fan of making things. Everything from knives to balms. I make all kinds of fishing lures. And I'm currently teaching myself how to do computer-aided design. First, I'm building a bird cage for a handicapped bird. But what I really like about this is that I'm going to be able to help nursery or greenhouse operations lay out schematics so they could see what it would look like at their operations, as well as, developing different machineries or, using my conveyor belt background to kind of, help visualize some of these more mechanization trends that are going in the nursery production world. I'm also a musician. My idea of a family get-together is playing music, and this has really instilled the sense of community and how important that is. And when many hands make short work, that's something I've embraced throughout my life. And I'm a big fan of the outdoors. I love fly fishing, being out on the water, into backpacking. A lot of my love of plants and fungi comes from the foraging side of things, and just understanding the world that's around you at any given environmental setting. So that being said, I've got a pretty rich diversity of information floating around in my head, and I think that's important not just for me but for everybody that's trying to solve a problem. The more angles that you can look at something, the more comprehensive your ideas can be. So next we're going to talk about my previous research for my Ph.D. The title of my dissertation was Blueberry and Cranberry Floral Stimulation of Calototricum fiorinea and Other Fruit Rotting Fungi. My major advisor was Dr. Peter Adamans, who's based out of the Blueberry and Cranberry Research Center down in Chatsworth, New Jersey. He is a Rutgers faculty. So with this project, it turned into a very large project very quickly. The reason we decided to work with Calototricum is that it's the main causal organism of blueberry anthracnose, and it's also one of the pathogens that cause cranberry fruit rot. It's known as a fruit rot complex. This is a very unique pathogen. It's able to form latent infections, and in the next slide I'll show you what a latent infection means. But more importantly, this pathogen drives the fungicide regime. So any fungicide applications are somehow in relation to this pathogen's presence in both blueberry and cranberry, and many other systems for that matter. Not only does it dictate the fungicide regime, but if you miss the timing, you could have upwards of 100% crop loss, so full crop failure due to this pathogen. So what is a latent infection? So typically, here we have the plant cuticle, we have the epidermal tissue, and this is supposed to signify a droplet from either a rain event or a wetting period, something like an irrigation event. And what happens in a lot of those times is that conidia also known as spores, are moved around in these water droplets, and they get thrown throughout the canopy. And when they land, the conidia put down an adhesion pad. They begin to germinate, much like a seed begins to germinate. And then the conidia can form secondary conidia, and these go on to cause other infections, or if this current infection is not successful, it's kind of a safeguard for the pathogen. And it also forms an infection structure, and this is the most important part of all of this, is that infection structure is able to punch down into the epidermal tissue, and basically cause a biotrophic infection. So at this point in time, this plant is infected, but the grower would have no idea that this is actually happening, because the pathogen begins to grow and grow and grow throughout the plant tissue until a certain trigger is flipped. Usually that's a conversions from acids to sugars, and the pathogen then goes into a necrotrophic colonization. At this point in time, you have symptoms, and from there it goes and it replicates again. So this hemibiotrophic latent infection process is really important, because there's a number of timing elements that you need to get right, or controls are not going to be successful. So why are we focused on this bloom period? Earlier, prior to me jumping into this project, there were a number of spray applications done to figure out when the best time to apply fungicide was. And so if you focus here on this red line, that's the percent in bloom of flower, percent in bloom. And above that is a staggered treatment regimen. So T1 is sprayed here, and then 2 weeks later. Then a week after the first treatment, a different treatment is started. So basically what we were able to see from this trial is that if you look at the T1 spray applications compared to the amount of disease in that earliest application, you got the highest level of control. Going up this scale is more and more and more fruit rot. So what this shows us is that there's a critical disease control window, and the longer you wait during the bloom period, the more infection you are going to get. So why is the bloom period so important? What are some of the mechanisms that are happening during the bloom period? But first we need to understand really why it's so important to have a proper timing. Because if you miss the timing, you're losing money in multiple different locations. Not only will you have a damaged product at the end, whether this be fruit or leaves or an ornamental to a fruit product, you're going to have a damaged product. But you're also losing money on the fungicide. Some of the fungicides are incredibly expensive, so they cost money. If you spend the time, you buy the fungicide, and you go out and you put down this material, then you're losing money on the cost for labor, the cost for the machinery, and the time that it takes to just get out there and do that. So the importance of understanding this timing event is critical, not just in blueberry and cranberry, but this is something that will be expanded upon in much more of my research, is to find that sweet spot where we can have good control without too much material, and we're not losing money in all these different components. So the reason we looked at flowers, and my main hypothesis here, was that plant signals produced during bloom play a critical role in the infection process and sporulation or spread of many of these fungi. Particularly, we looked at blueberry and cranberry, but this is also something that you will see throughout this talk that comes up. Just wanted to show you real quick so you know what I'm talking about. These are blueberry flowers, and these are cranberry flowers. They're wildly different looking plant species, but they're highly related. So much to the point that some of these can be bred together. So with this project, there were two main questions that we had to address. First is, how do we isolate a floral signal, which turned out to be fairly easy? And then how do we evaluate the pathogen response to this plant stimulus? So first, and I'm not going to go into a ton of detail because we did a video methods and I'll talk about that later on, but very simply, we had two different types of extractions. We had water-based and chloroform-based extractions in which we mixed it. We mixed it for 30 seconds, strained out the floral material, and then we're left with the floral extracts. So that is the floral signals, either trapped in water or trapped in chloroform. From there, we developed a glass bioassay. This is an adaptation of a previous glass bioassay, but we kind of took it up a notch to expand upon it. So within the bioassay, there are two main treatment groups. There is the flower extract, also could be considered the plant stimulus treatment, that has water, the stimulus, and the fungal material, which is then compared to a water control. You take droplets of these, place them onto glass cover slips, you let the fungus grow in the presence of the floral signal, and then at a predetermined time period, like anywhere from 2 hours to 12 hours to 24 hours, you stain it with lactophenol cotton blue. You perform a 4-field analysis on it, and then you have the distinct pleasure of counting thousands upon thousands of spores and infection structures. But this is really cool because this takes it from being just a qualitative test to a quantitative, where we can put real numbers associated with how this pathogen is responding to a plant stimulus. So just to give you an idea of what that looks like when you compare those two different treatment groups, here this is a water extract, and every once in a while you'll have a replication event, or you'll have the formation of an aprosoria with the infection structures. But when you mix this same load of fungi with a floral stimulus, you have magnitudes more stimulation. So both of these treatments started off as the same initial concentration of conidia, but after a 24-hour period, this is magnitudes larger. These are all secondary conidia, and these are the infection structures. So like I said before, cranberry fruit rot is a complex of different pathogens, and we wanted to know if Calototricum was the only pathogen that picked up on this floral cue. So we looked at different groups of cranberry fruit rot pathogens. Here, the one we've been studying the most is C. fioriniae, but we also looked at these other groups that are from different genera and families, for that matter. And what we found from this is that the pathogens that are most adequately controlled during the bloom period were the most stimulated by the floral extracts. So showing that there was a legitimate interaction between the fungi and a host stimulus produced by the plant. So I don't have time to go through all of the different pathogens, but generally speaking, the outcome of this first wave of the research was that floral signals stimulate sporulation events or secondary conidia, and they also increase the number of infection structures, the aprosoria. But how these flowers affect the epidemiology of fruit rot is really important. And that's the part that we can key in on to actually have make a real difference in the field with this type of information. So floral extracts. So here we have hours post inoculation. These are the conidia, these right here. And what you can see in the presence of floral extracts, this red line here, as you allow the fungi to be in the presence of the floral signals, you have increased levels of conidiation events that really become significantly different in this 12 to 18 hour time frame. So that means that floral extracts are increasing inoculum, thereby they can increase the spread of disease. Next, if we look at the infection structure or the aprosoria, looking at that same time scale, you have significant differences at 6 hours, meaning that aprosoria are forming in much less time in the presence of flowers, so infection can happen that much faster. So if you expand this out into a field setting, if you think about a wetness period in the field that is 16 hours long versus a wetness period in the field that is 6 hours long, you're going to have many more 6-hour wetness periods. So during the bloom period, there's going to be a greater potential for disease. Not only do they shorten the amount of time needed for these biological activities to occur, they also influence how the pathogen behaves at different temperatures. So here we have total conidia and aprosoria again, but in the presence of flowers, at lower temperatures, you're getting, albeit slight, you're still getting greater levels of infection or the aprosoria forming. And this pretty much holds through the rest of the time points, but if you have a shorter infection period and a lower temperature range, the probability of having an infection is that much higher. Additionally, as the temperatures begin to increase, you get more and more and more secondary conidia produced. So when the temperatures get warmer, the disease is spreading. So if we take those two components, we can use them as epidemiological factors, such as the time and temperature, and feed them into future disease forecasting models. And as an example here, NIWA. NIWA is based out of Cornell, and it has a series of different modeling programs that look at different diseases. So we think with the additional floral information, we can better inform a lot of these disease forecasting models. So in conclusion for this section is that floral signals decrease the time needed for infections to occur, as well as sporulation events, and they alter the temperature range of bioactivity. Some of the other components I don't have time to get into right now is that there seems to be potential cultivar effects in blueberry where susceptible cultivars are more stimulatory, and the floral extracts can also be used as a tool to complete Koch's postulates, which is notoriously difficult in cranberry pathology. Another really interesting component of this research too is the field monitoring. I developed some rainwater collection devices that basically captured rainwater that had run off of flowers and tracked these floral signals or the host signals throughout the whole season. And what we found is that floral signals are mobile, meaning they can activate multiple inoculum reservoirs, as well as they change over the season, where the plant signals coming off during the bloom period are way more stimulatory than signals that are coming off from developing fruit. So I hope at this point in time you're asking yourself, what are the plant signals? This wave of the research could be expanded out into many more years and many more PhDs, but just to give you an idea of what the floral signals are, this is a epicuticular layer. This is basically the surface of a flower or a fruit. Any plant part has this basic design. You have two wax films that are composed of C16 and C18 hydrocarbons, and from them you have these wax and polysaccharide crystals extruded from the top of the surface that give it this kind of geometric shape. These molecules extruding from the surface are more of those C16 and C18 hydrocarbons that are fatty acid derivatives, and they're used to make all kinds of molecules. But for this research, for this demonstration, we're just going to really talk about the lipids, the hydrocarbons. What we did find looking at the sugar side of things, or the nutritional side of things, is that sugars are very much involved in sporulation events because they're highly nutritious, allowing the pathogen to replicate using that energy source from the flowers. So looking at the fatty acid skeletons, we knew that the materials extruding from that surface in their simplest form were fatty acid derivatives. So we utilized what's called a GC FAME technique. And we collected samples from multiple blueberry and cranberry phenology, different cultivars, different extraction types, both the chloroform and the water extractions. with a total of 116 different samples characterized in triplicate. I was locked in a dungeon for quite a long time trying to figure this all out. But at the end, we got this heat map. So the reason I decided to use these heat maps is it shows you the different fatty acids, their relative abundance in all of the different extraction types. And looking through, there were different compounds that were more closely associated with stimulation. And through a multitude of experiments, we identified that hexadecanoic acid, one of those original C16 hydrocarbon, one of its derivatives, which is a component of cutin, was the most stimulatory. This wax is incorporated into all kinds of other structures on the surface of plants. And it also makes sense that a fungus would pick up on a derivative of this compound, because many plant pathogens excrete cutinase. And cutinase dissolves these C16 and C18 hydrocarbons, and it feeds into this loop. So once the pathogen understands where it is on the surface of a plant, it will begin to try to infect it. As you can see it here in the little yellow circles here, these are infection structures that have formed in the presence of this compound, compared to a chloroform-only treatment where the pathogen grew, but it didn't go into this strong infection structure stimulation. So to wrap up 8 years of my life, basically what we found is that the hex compound triggers aprosorial formation. Different sugars, particularly sucrose, stimulate conidiation events, and that can explain some of the bioactivity that we're seeing with the floral extracts. Is this the whole thing? No way. There's much more to be investigated, but it's a great starting point or foundation for future research, which up until this point in time hadn't really been looked at all. So what we discovered, we didn't just discover that these molecules exist and what they do, but with this information, we can use that, we can weaponize that. What we can do is use some of these compounds to stimulate plant pathogens in the presence of fungicides or other adverse conditions, such as disrupting host recognition. By using these compounds to kind of trick the pathogen in thinking it's a different time of the season. And this was developed in blueberry and cranberry, but this whole concept of modifying host surfaces is something that I think can be incredibly useful in the nursery and greenhouse industries, with different adjuvants, different wax applications, where we could get a little bit better disease control with using less materials. For a multitude of reasons, that's a benefit. And the bioassay that we developed to do all this research is not only just to deal with blueberry and cranberry. We can use root exudates to deal with Phytophthora. We can collect waxes to deal with botrytis. There are so many different things we can use this bioassay for, and an idea that I have we're going to talk about in a little bit. But the slide that I like to bring home this whole idea of this project is that if floral signals decrease the time needed for infections to occur and at lower temperatures during the bloom period, then there are inherently going to be more infection periods during bloom than there will be at other times of the year. So this means you have to focus your attention to this period so you have a product at the end of the season. So went through, this research is really dense. There's a lot of really good information that we put out. We developed a video method, so if this is something you're interested as research, or you have an idea or something that you want to talk about, go ahead over and check out this video. We made it open access so anyone can see it. We also produced 2 papers in phytopathology, which is the seminal research group for phytopathology, in both blueberry and cranberry. But there's slight differences in methodology. and feel free to ask me about them as well, or some adaptation of that research. So that's the end of my dissertation research, but when I started my internship at the Marucci Center, I was given a project that had been kind of stale for a while because no one could figure out how these pathogens were being spread. So I looked at what is called sooty blotch and fly speck of blueberry, and this was a great entrance for me into the world of fungal pathology. And so what we figured out is that I went out to the field one day during a rain event, and I was looking at where the water droplets collected and where the prevalence of the pathogens were, or this black covering here. That's the sooty blotch, and then the black dots are the fly speck. And through that, we were able to complete Koch's postulates. And we also identified over 70 different fungi that cause this complex. And with that information, we were able to give an idea of what's going on to a lot of the blueberries that were concerned about it. I was asked to give a presentation at Hershey. My presentation got picked up by the Fruit Grower News, and ultimately, because this was a new wave of research, our research got added to the compendium of blueberry and cranberry diseases, as well as using this information to gather larger grants to hire postdocs or other researchers. So this was a really key thing for my diagnostics, knowing what I'm looking at in the lab and helping me figure out what the pathogen is or problem is. Is it a fungi? Is it a bacteria? Or is this a viral symptom? And that's one of the things I hope to bring to the nursery world as one of my skill sets in pathology. So you might be thinking, so this guy has done a lot of work in the lab. Why does he want to be an extension agent? Well, I did do a lot of work in the lab, and that was a lot of long nights and 40-hour work days. But I've been out in the field, and I've designed and implemented all kinds of different spray trials. Specifically, did blueberry, cranberry, and grape. We looked at regime trials, cultivar susceptibility trials, the weaponization of those floral signals, as well as I wrote and got a SARE grant looking at some bacteria that I discovered on grape flowers and adapted that to use on the blueberry system. And then we compared that to different biological controls. So when it comes to designing and implementing and applying, for that matter, spray trials, I've done a lot of it, and that's something I definitely plan on using in my plant health side of my nursery extension program in the future. So moving forward, I started January 1st, and with everything that's going on with COVID-19, it's been a very challenging and interesting entrance into a new career. But I have been able to identify some needs, and please, I'm always available if there's something you think I should be looking at. But with a nod to my past, I don't plan on bringing blueberry spray materials over, but there are definitely some things that I think our industry, our nursery industry, could really benefit from. Like I stated in the beginning, I'm now with Rutgers Cooperative Extension in Cumberland County. I'm dealing with most of the southern state or more of the commercial nursery side of things, and I'm really happy to be there. It's a great office and great people to be working with. Good team. So in blueberry production, there are a series of charts that go into when you want to spray for different diseases according to phenology or degree days. And this is something that we can't make one list for every plant that is in nursery production, but what we could do is break down lists like this for different families. Like ericaceous plants is a great place to start because it's a lot of the big money-making plants. We could also break it down into different groups like conifers, different herbaceous perennial subgroups, ornamental fruits. And we can also create these on prevalence, like market need, where boxwood, hollies, Christmas trees, crepe myrtles, everything that is commonly grown and it's a staple, we could develop specific charts for that. So the idea being that you could grab one piece of paper and it'll tell you what diseases you should be looking at this point in time, kind of help bring more efficiency to our, that's the next point, more efficiency per unit time, because labor is a big deal. So if we can hit more things in one fell swoop, all the better. Additionally, so this graphic is coming from grape, and it goes into all the different fungicides that are available for a number of different diseases. And there are two main take-homes from this. And it also gives the risk group of all these different fungicides, because fungicide rotation is important. But what it shows there is that you have the importance of chemical stewardship, that you need to rotate high-risk materials with low-risk materials, but not all of the compounds actually work. We know this. Some compounds work better than others, and this gives us the opportunity to use both of those components. That this compound works really well, but it's high risk, and we have to rotate it with something that maybe doesn't work as well. But it would be great if we could speed up our understanding of these different pathogens, or of these different materials, as far as which ones worked and which didn't. So to make every time you're going out there and doing this count, with the idea being is that the bioassay that I was talking about previously in the presentation, I also adapted it to work in 96 well plates, where we could evaluate different compounds with different host signals and with different diseases. So the way that would look is that, across the columns, we could test different rates, new materials. We could test compounds that didn't work so well, or compounds that we think are going to be good in the future, with host stimulants such as root extracts for Phytophthora studies. We can also compare a lot of these different compounds to a number of different pathogens at one time, because we want to make this count. And this is something that can be done relatively quickly. In the assay run takes about 12 hours to 18 hours to run, and maybe another 12 hours to get full data on. But in a day, we can predict what a field trial is going to look like. This doesn't replace field trials, but this gives me and the recommendations that I hope to create an advantage on being able to screen more materials quickly, because there is a finite amount of times that anybody can be out there spraying materials. So anything we can do to improve that will be great. So I have some examples of slides that I would give when teaching undergraduates or classes looking at the specific biology of different pathogens. Phytophthora and different oomycetes like Pythium and downy mildews are very important to the nursery industry, and that's somewhere that I definitely plan to spend some focus on. But like I've alluded to a few times, this is a perfect opportunity to mesh my previous work experience with ideas moving forward. And generally speaking, this pathogen, I want to develop better nursery-oriented best management practices for irrigation lines. I want to look at biofilm accumulation and how that impacts root rot diseases. Look for developed tools that the growers can use on site that will give them an indication of whether or not there's a Phytophthora bloom going on in their irrigation or their recirculation water, something that's quick and visual and cheap. Another area that's problematic and important to continue to look at are bacterial diseases. We need more than copper, and there's a limited set of antibiotics. That being said, copper is a low-risk group, but it's high risk in a lot of other ways, especially in the propagation house. If it's going to burn roots, it can be a particularly hot compound. So continued research with this, looking at more biological oriented control mechanisms, I think is going to be something really important. And bacteria can also be used in that bioassay that I'm, continuing to develop here. Another area of research that I'm very keen to learn more about, admittedly, I'm learning every day more about entomology, but looking at insect vectors. Insect vectors is a great way to control diseases that you can't see until it's too late. And like I said, this is a perfect entrance for me into entomology because I know it's so important to the nursery world, and I want to have a solid understanding so I can be there to help for this. Think of some things that maybe haven't been thought of before. Who knows? Another thing that I like to bring up pretty routinely in grower conferences is the idea of that we do not want pesticide resistance. We don't want it. It happens and we don't want it to happen. And it all really boils down to one simple thing, is that this is one topic that if we go about it correctly, we can make our lives easier. This is guarding something that's helping us. Because when we lose a compound, in a lot of cases, it's gone. When we lose a compound, when we push any type of pesticide regime too hard on any given production area, we can have localized populations that are immune. And that does you no good if someone else isn't immune to it. If the ones that are living on your farm or nursery or greenhouse are immune, you got to find a different chemistry. And I like this quote a lot. Jason Slaught from Ohio State, and just remember, he's talking about copper here, a low resistance material. The overuse of anything is a bad idea. And this makes sense because we do only have a few limited things for certain diseases, but we need to continue and try at least to find different approaches, different cultural practices to combat these different issues. And just drive home the fact that pesticide resistance happens. It's very real. It happens in insects, weeds, plants, mammals, you name it, it happens. So the idea of chemical stewardship is really important, and it's something relatively easy we can do in a lot of cases to not make our lives harder. The other component that I hope to bring in my educational part of the extension is I like the idea of growers understanding a little bit about the biology, not just that I spray X, Y, and Z. And I know I view growers' intellect of their specific locations and how different crops work and how different pests work an extremely high level. So I hope to learn more of the practical biology components of those systems. Because going into pest biology or how a monoculture actually impacts pest dispersal and very importantly, how human practices impact everything on a farm or nursery is really key. I like the analogy of you don't go to class to learn what's in the book. You go to class to learn about things that are kind of alluded to in the book, but aren't necessarily there. You are going there to learn tools of how to understand situations sight unseen. So armed with that knowledge, this type of scenario could be present. In a lot of ways, and in a lot of cases, this is a great way to water a lot of material really quickly. However, if you know that this specific area is prone to phytophthora infections, or there's a particular pathogen that should be sporulating at this time of year, armed with the knowledge of basic biology, this might actually be the worst thing you could be doing at that point in time. It might be the perfect way to spread disease. So having the concept of people feeling comfortable and making good decisions based on biology is something that I really hope to bring to my extension program because it's important and it can save us a lot of time and money. Some of the other pest issues that I've identified, and like I said, if you have suggestions, ideas, criticisms, you name it, please contact me. My email is at the end. I'm very open to, I just need to know what the questions are so I can focus on them. Some of the things that I've picked up so far are boxwood blight, in addition to treatment options. Also, best management practices for quarantine issues. How do we set up a farm so if we do have an event happen, how do we navigate that in the most logical way possible? Like I said before, bacterial diseases and Phytophthora, focused on developing best management practices, fact sheets, recommendations, real hard copy recommendations that also deal with cultural control elements. Flea beetle, ambrosia, and whatever invasive species is currently on the menu. I'm very keen to learn more about the biology of these systems and identify vulnerable periods and how we can attack these pathogens in a more critical way that makes every spray application or every unit labor count for more. Weeds is also something that is notoriously difficult to deal with in containerized nursery production, or all of nursery production for that matter. And I'm keen to use my spray background to screen different herbicides and look at different over-the-top herbicide applications, or kind of try to think a little outside the box here and work with our weed specialists here at Rutgers to get some momentum going on weed control in the nursery and greenhouse settings. Additionally, looking at all these pests and diseases, I'm really keen to develop a localized or a Rutgers website-based degree day calculator. I'm thinking in terms of an app where you type in your location, what date you want to look at, and it generates a degree day, but then it also tells you what pests are going to be a problem at that point in time, as well as links to different fact sheets or what materials you should be thinking about having on hand in the future, as well as what time of year you want to be out and scouting. And this website-based idea goes hand in hand with the charts that we talked about previously that I'd like to develop. Idea being that you can come in and leave a Rutgers website armed with much more information than you came in with. And speaking about information, one thing I'm keen to really trying to focus on since there's been the new limited engagement due to COVID-19 is I've been really working on trying to get the Sustainable Nursery Production webpage up to speed. This was started by Sal Mangiafico, and he did a great job, and he's kind of handed the reins off to me. We're going to use that as a central repository for links to different important websites, fact sheets, as well as links to videos, especially at Rutgers demonstration areas. In the process of acquiring materials to get some high tunnel and pot production areas set up over at Rarick, as well as access to forms that everybody needs, and it's just a place you can go and print them out. And another component that I'm pleading to the growers with here is you guys have great information. You have such incredible practical knowledge. I think a grower show and tell would be really useful. In the cranberry industry, they did a grower show and tell at every one of their field days. And it was just a great idea to look at new machinery. And it's something similar to a Twilight meeting, but if somebody had a great idea and they didn't mind sharing, I'd love to make a video of it and, post it so more people can learn. And I haven't mentioned Bill Erickson of Monmouth County was also recently hired, and he and I are more or less the nursery extension agents in the state right now. And so cooperation between us is really important to us because we want to show a unified front that any issues that you're having in nursery production, we're there to help. On that same idea, we're reinvigorating the Rutgers Nursery Working Group with my expertise in pathologies, Bill's expertise in soils, Raul's in physiological, Sal has water components, Steve's insight into IPM and Hamont's insight into mechanization, between all of us and other people that we bring in for specific tasks, we're really excited that we're going to be able to crank out some good information and, like I said before, hard copy recommendations, as well as cooperation between academic, chemical companies, and regulatory. We are all doing similar topics, but we're all definitely on the same team. So cooperation there is only going to help the growers, as well as breaking the language barriers. I currently am taking about an hour a day Spanish lessons in an attempt to learn Spanish better, at least on a speaking level, because multiple people have asked for more basic education, basic biology or mathematics education related to agriculture in Spanish, as well as a question to the growers is, do we push for Spanish licensing exams? Because let's face it, Spanish is a language that is important to our industry. So I'm interested in any way I can to help make that more accessible, knowledge more accessible to those people. So in conclusion, these are the acknowledgments from my previous life. I've worked with SARE before, the Cranberry Growers Association, Blueberry and Cranberry Research and Extension. And I must say that without the Haynes family, who supports the William S. Haynes Senior Endowed Cranberry Research Fund, it is very unlikely I would be sitting here giving this presentation right now. They allowed my dream of being an extension agent to come to life, and for that, I'm eternally grateful. I did get my Ph.D. through the Department of Plant Biology. My advisors were Dr. Peter Ottomans, as well as Brad Hillman, Donald Kobayashi, and James Polishak at the USDA ARS. But I'm really excited too that I can add new interactions to my acknowledgements list, especially these grower groups that have been very great in helping me spread information or keep me up to date on different topics, especially during this very intensive COVID information overload period. So with that, I'm really excited to be here. I want to help. I want to help for real reasons. Like I said, my friends are growers, and whatever you need, I'll try to get you the answer. As a new agent, I might not have the answer right away, but I am good at looking up information and getting back to people. So that's my my email there, and thank you very much. Also, yeah, make sure you check out the Plant and Pest Advisory, the LONT edition for landscape and nursery, as well as the Sustainable Nursery Production page. Again, my name is Tim Waller out of the Cumberland County Extension Office, and thank you very much for your attention.